Hello, everyone. Thank you for attending the first lecture of the Scientific Journal Club of GESR. I would like to introduce Dr. Mohamed Gamal. He is a doctor of, he was working here in Germany, and he got his doctorate title from the Charité Universitätsmedizin Berlin. And he also currently is a lecturer in the University of Zakazig in Egypt. So thank you so much for giving this lecture, Dr. Mohamed, and we are happy to hear you talk about this topic. Thank you.
Many thanks for you, Dr. Ahmed, Dr. Nurhan, Dr. Khalid, for this great organization to give me the honor to be the first presenter for the Scientific Journal Club belonging to the GESR. I would like also to welcome Dr. Dema Adwan. She is Dean of Faculty of Medicine. I am very honored to have you today among our speaker, and she is a consultant also of gynecology and obstetrics. So we have today unexpectedly an international scientific journal club. I think we are very, very proud to have this international audience today. So I will try to talk today about my own scientific research. I think I have to get my PowerPoint presentation first uploaded here for the Zoom. Yes, this is the one. Can you see the slides?
Yes.
Okay, good. So this is a presentation which I had to give to get my doctoral degree at the Charité University of Medicine a few years ago, and from these studies, from these results, I have also published 3 main publications in international peer-reviewed journals, and the topic was endometriosis and adenomyosis. The topic is, or the title of the talk, New Insights into the Pathogenesis of Adenomyosis, with a special question: is the dislocation of the endometrial fragments into the myometrium evident in adenomyosis uteri? So as I see, you are not all a gynecologist, so I will try to define first what is endometriosis. So each woman during her young ages, beginning at 12 and ending at 45, maybe 50 years of old, they are getting their menstruation, their periods, their menstrual period every 4 weeks almost. The menstrual period or the menstruation or the menstrual blood, this is formed by the inner layer of the uterus inside the belly of the woman. But there is some women who are suffering from a condition called endometriosis, and this is the endometrium. Endometriosis is a condition of the occurrence of endometriosis, but outside the uterus. So you find here endometriotic lesions on the fallopian tube, on the ovary, on the peritoneum, inside the pelvic cavity or inside the abdominal cavity. This is what we are calling endometriosis. It's a pathology. It's a disease and characterized by lesions which are active, hormone dependent. They are menstruating as well every 4 weeks. So patients with endometriosis usually have also bleeding inside the abdomen, but this is a minimal bleeding, leading to irritation of the peritoneal cavity of the nerve system. This leads to much pain for these endometriosis patients. Okay, there is a special type of this endometriosis, what's called adenomyosis, and this is actually the topic of today, adenomyosis. Adenomyosis, this is a condition in which we find these endometrial lesions or these endometrial tissues, but inside the muscle layer of the uterus. Usually the uterus has 2 layers. The inner layer, this is the endometrium, which is usually sloughed during the menstruation, during the occurrence of the period, and comes out during the menstruation to outside. But sometimes you have endometriotic lesions or tissues inside the muscle layer of the uterus, and the muscle layer of the uterus called myometrium. And this is something irritating the muscular power or the muscular layer of the uterus and lead to much pain during the menstruation. And this is what's called adenomyosis. Actually, there are a lot of theories which are trying to explain the occurrence of adenomyosis. And one of this theory, the recent one, the tissue injury and repair theory. It's supposed that this is the uterus, this is the endometrial layer, and this is the muscle layer. And it's supposed during the menstruation, there is a lot of contraction of the uterus trying to expel the endometrial, the sloughed endometrium, the sloughed inner layer of the uterus to the outside. But in these patients with adenomyosis, we have some sort of injury, of tissue injury between two layers. Between the endometrium layer, this is the inner layer, and the outer layer, the muscular layer. And this forces the endometrial tissues inside the muscular layer of the uterus and leads to the occurrence of adenomyosis through a process of prostaglandin formation or prostaglandin synthesis accompanied by some hormonal changes. Not going in details in this slide, but just to know tissue injury. We have some sort of tissue injury occurring during the menstruation because of the contraction of the uterus. And this is usually followed by a trial of repair by the tissue, by the uterus, to repair this injury. And when you have a failed repair mechanism, you get the adenomyosis. Yeah? So you have established endometriotic lesions inside the muscle layer of the uterus. Good. So this is an animation. This is the upper layer. This is the endometrium. This is the myometrium. And due to an abnormal contraction of the uterus, you see here some forced endometrial tissues. So this is a glandular tissue again inside the myometrium of the uterus. And these tissues are going at some sort of transformation and establish the occurrence of endometrial glands, but outside the endometrial cavity. So inside the myometrium, this is ectopic endometrial tissues, which we are calling adenomyosis. I hope it's now clear. Adenomyosis is a condition in which we find ectopic endometrial tissue inside the muscle layer of the uterus. So we are speaking about active endometrial tissue glands, but in an abnormal site. This is the muscular layer. This is the myometrium. A second point, which is called the myofibroblast and the tissue trauma. We have fibroblast, and all of us are familiar with the fibroblast. Fibroblast cells, specialized cells producing collagen, and they are involved in the process of repair. But these fibroblasts usually activated by a mechanical tension or by a tissue injury, for example, and they get some special characters to build smooth muscle-like bundles inside the fibroblast. So you have some sort of a combination of a fibroblast and a smooth muscle cell. How can we imagine it? The fibroblast, this is just like the factory producing the collagen cells. But if this fibroblast got this smooth muscle-like character, so they are considered like mobile factories. Yeah, so they can move to the site of tissue injury, and they can settle up, and they can produce the collagen to share in the repair process at the tissue trauma, or at the site of tissue trauma. This is what we are calling myofibroblast. The important message of this introduction to say, whenever you see myofibroblast inside a tissue, so this is an indirect evidence of tissue trauma. And this is actually, this was so the two main research questions in my doctoral work. The first one, in adenomyosis patient, do we have any evidence, either microscopic under the light microscopy or ultra microscopic using the transmission electron microscopy of tissue trauma at the junction between the endometrium and the myometrium in adenomyosis patient? This is the endometrial myometrial junctional zone. This is the junctional zone between the endometrium, the inner layer, and the myometrial muscular layer. Do we have any evidence, microscopic or ultra microscopic evidence? And the second research question, if there is any microscopic or ultra microscopic evidence of translocation of these endometrial glands inside the myometrium of the uterus. So we started our study. This was something like 2011, 2012. I have collected the samples prospective because I had to examine them by the transmission electron microscopy at the end. And transmission electron microscopy, you need fresh samples to be prepared differently from the tissues which are used to be examined by immunohistochemistry or by histopathology. So I collected young patient, underwent hysterectomy, so the removal of the uterus, and this is something not common. Yeah? This is a first difficulty to have young patient get their uterus excised. So the number here of the sample size was not so big, but it's actually a big number for such a study design, yeah? Because we relied on young patient underwent hysterectomy, and we had to exclude or to confirm the adenomyosis in these uteri first. So the uterus have to be examined firstly if the uterus has an adenomyosis or not, to split them into a case. In a case group, this is the adenomyosis group, and we included 18 women, and non-adenomyosis group, this is the control group, and this was 14 women. And we proceeded further to a transmission electron microscopy, immune histochemistry, immune fluorescence, Van Gieson staining. I am going through the next slides. The second challenge was where should we get the samples from the uterus? We have a uterus, it's weighing about 100 grams, 70 to 100 grams, and dimensions about 7 by 5 by 3 centimeter. So we have to standardize the sample, the taking of the sample or the sample biopsy from the excised uteri. So I developed this diagram to be a standard. This is the fundus of the uterus, this is the body of the uterus, this is the cervix, almost one third, and this is the middle 1/3, and inserting a uterine sound inside. So I could identify the fundus to the upper limit of the uterus, and through a cutting line here, I split the uterus into 2 pieces. And from the anterior wall and from the posterior wall, I took a wedge biopsy. Yeah? The wedge biopsy assured us to have both the inner layer of the endometrium, so the inner layer, this is the endometrium, and the outer layer of the uterus, this is the myometrium. And this wedge biopsy was again splitted into 2 processes. The first process for a transmission electron microscopy examination, and the second process for immunohistochemistry and immune fluorescence examination. So this was a second challenge, how to collect the specimen from the excised uterus. Now going further to the results. The first research question, do we have any evidence of tissue injury in the uterus? And surprisingly, yes, we have seen microscopic evidence of tissue injury. So to imagine it, this is a non-adenomyosis patient, and we have, this is the endometrium. This is the upper left one from the photo here. And this is the myometrium. And if you see, the junctional zone here is a straight one, not interrupted. Compare it with the last slide here on the adenomyosis on the right side. You have this endometrial gland, this is the endometrium gland, this is the myometrium, but you have lost here, you don't see any regular line between the endometrium and the myometrium. And this is what you can see here. This is the endometrium again. And if you see the junctional zone, this is the junctional zone. And then you see a dipping down from the endometrial gland inside the myometrium, as if the endometrial myometrial junction zone or interface is quite fissured. So we have fissuring in this junctional zone between the endometrium and myometrium. And this was the first microscopic evidence of occurrence of tissue injury in adenomyosis patient, but lacking this evidence, was lacked in the non-adenomyosis patient. Again, the same we have seen also with a transmission electron microscopy. If you would like to see here, this is the smooth muscle cell, a fusiform spindle-shaped cells. This is the nucleus. This is the second one. This is the third one. They are all parallel, arranged to each other. So they have a parallel arrangement. But if you see here, you see the smooth muscle cells again. This is in parallel arrangement. but another group bundles of smooth muscle cells, and they are taken in a cross section. So they are arranged in a different direction. So in this slide section on the right side, you see smooth muscle cells arranged in different directions. On the left side, you see smooth muscle cell arranged parallel to each other. The loss of this parallel arrangement of the smooth muscle cell, it's again an finding or an evidence of occurrence of tissue injury at the endometrial junction zone. Proceeding further with the immunohistochemistry, we used the collagen antibodies to stain the inner myometrium of the uterus. As all you know, the smooth muscle layer, this is the myometrium, usually lacking collagen. Usually you don't have much collagen inside the inner myometrium because this is a muscle layer. This is a smooth muscle cells. But interestingly, here you see a lot of red stained areas, and this is a lot of collagen producing smooth muscle cell. This is back again to our first slide in the introduction. Collagen producing, this is a character of fibroblast, but in a smooth muscle cell, so this is what we call myofibroblast. So the smooth muscle cell have changed to another form of myofibroblast, producing collagen and still having the smooth muscle cell criteria. And this is again an evidence of tissue trauma at the junctional zone between the endometrium and between the myometrium in adenomyosis patient. A further finding or evidence of occurrence of tissue injury is that we have alpha smooth muscle actin. This is a marker, a protein usually produced by smooth muscle cell. But if we see here, this is the endometrium glands, and the endometrium glands in a non-adenomyosis patient, usually there is a quiet environment. There is no tissue injury at this level, so you don't see any labeling for this marker, for this alpha smooth muscle actin marker, because you don't have a smooth muscle cells, except for the small blood vessels. Inside the wall of the small blood vessels, you have smooth muscle cells, and then they will give you the red staining. But compared to the adenomyosis, you see the difference. This is again the endometrial glands, but surrounded by a lot of endometrial cells, but stained with alpha smooth muscle actin. The endometrial cells usually are some sort of fibroblast, but now they gain the smooth muscle criteria. This is the reverse from the previous slide. Here was the smooth muscle cell gained the fibroblast character. Here is the stroma fibroblast cells gain the smooth muscle character. So again, we have a lot of myofibroblast inside the endometrial layer in adenomyosis patient, and this is again an indirect evidence of tissue trauma at the level of endometrial myometrial junction zones. Very, very interesting was also on the finding of transmission electron microscopy. This is an endometrial gland, and this is the basal layer of the endometrial gland. This is the nucleus, and this is the epithelial cells. So we have one nucleus, second nucleus, third, fourth, fifth, sixth, seven. So seven nuclei with 7 epithelial cells arranged parallel to each other, looking very regular oval on non-adenomyosis patient. When compared to the adenomyosis patient, you see the nucleus is looking heterogeneous. So you see there is an enfolding of the nuclear membrane of the endometrial glands. Inside the endometrial glands, inside the endometrial epithelial cells, you see an enfolding, many enfoldings in the nuclear membrane of these epithelial cells. And this may be an indirect evidence of tissue trauma occurring at the endometrial myometrial junction zone. So this was the first answer for, or this was the answer for the first research question. Do we have any evidence of tissue trauma in adenomyosis patient? Yes, we have seen it both on microscopic level as well as on ultra microscopic level. That's to mean with the transmission electron microscopy. So proceeded to the second question. Do we have any evidence of translocation of these endometrial glands inside the myometrium? This is to simplify it. Do we have any dislocation of these endometrial glands due to this trauma in adenomyosis patient inside the myometrium? we couldn't see any evidence of translocation because, first of all, here with the ultra microscopic examination with the transmission electron microscopy, we see again here in non-adenomyosis patients, this is the nucleus, this is the nucleus, this is the nucleus, yeah, and this is the endometrial glands, the upper one. The lower one, this is the stroma, this is the outside the endometrial glands. And when you see here, this is the membrane between two epithelial cells. This is the middle one and on the right one. And if you look inside the circle, you see a black point. This is the desmosomes. Desmosomes, it's a form of cell-cell contact. And whenever you see this cell-cell contact, you can consider it as an evidence of an intact cell-cell contact and no evidence of dislocation or translocation of the basal endometrial glands inside the myometrium. Again, you see here the desmosomes. This is to say the epithelial cells are closely attached to each other, and there is no leakage between the cells inside the non-adenomyosis patient. What about the adenomyosis? In the adenomyosis, theoretically, you are searching for cells with much leakage between the cells, secondary to the trauma, which we have seen. Microscopic and anti-microscopic. Surprisingly, there was no evidence of loss of this desmosome cell contact between the epithelial cells, because again, this is the cell membrane between this middle epithelial cells and this epithelial cell on the right side, and you see the desmosomes. This is a very tight type of the cell-cell contact, and they are intact. Not down-regulated, so we can exclude now any sort of translocation or dislocation of the basal endometrial glands inside the myometrium. We have made also an immune labeling, immune histochemistry with the E-cadherin, and this is also a marker for adherence junction, a type of cell-cell contact again, and we haven't seen any difference. This is the endometrial glands stained with red, and the same intensity and the quantity of the stained cells were the same in both adenomyosis and non-adenomyosis patients. This is to exclude any form of translocation or dislocation of these endometrial glands inside the myometrium secondary to the trauma. So we, at this point, good, we have an answer. First, we have an evidence of trauma, but this evidence of trauma can't explain the occurrence of the endometrial glands, the ectopic endometrial glands inside the myometrial cells. So what is the cause of the occurrence of these ectopic endometrial glands? And this was a very interesting finding that we have seen. We have seen, this is again, so a diagram I have drawn myself. This is the endometrial glands. This is the epithelial cells, yeah, with tight junction inside. And this is the inside of the this is the lumen of the endometrial glands. This is the stromal cells. So we are speaking about the inner layer of the uterus. This is the endometrium. On the periphery of the endometrial gland, we see cells which are not stained. So I call them the pale cells. Pale cells, cells which are chromophobe, so didn't stain with any H&E staining, hematoxin and EZ staining. They haven't been, or they couldn't be stained. And this was frequently seen among different endometrial glands. What is unique in these glands, in these cells, these pale cells, that they were situated between the basal membrane of the endometrial gland and the epithelial cells. And to imagine it, this is again so a transmission electron microscopy. This is the endometrial gland. This is the basal membrane. And the whole slide here representing so the endometrial gland. This is the endometrial epithelial cells, and this is the endometrial lumen, or the lumen of the endometrial gland. And if you look here to the white arrows, you see these cells here inside. They are situated or located between the epithelial cells and between the basal membrane of the endometrial gland. This is the first one, this is the second one, this is the third one. So they are not epithelial cells because they don't stain with H&E or with other markers. So they remain pale, which we are called electrolucent. This is a magnification, I think it was about 10,000 magnification of these pale cells. You see this is the cell borders, a cytoplasm very rich in microorganelles with ribosomes, with Golgi apparatus, with mitochondria, and looking electrolucent. So this is a pale to the surrounding, compared to the surrounding epithelial cells. So to imagine these pale cells, you can imagine it as if the pale cells are hugged by two epithelial cells. So you have an epithelial cells on the left side, you have an epithelial cell on the right side, and then you have the pale cell hugged by two epithelial cells. These pale cells have very unique characters. First, they lack any form of desmosome, and desmosome, this is a type of cell-cell contact. So they are loosely attached to these epithelial cells. And this is what we have seen on real here at transmission electron microscopy. This is the nucleus of the pale cell. This is the cytoplasm of the pale cell. And this is spaces between the epithelial cells and between the pale cells. So you see in this location, on this side, there is no type of cell-cell contact. There is again no cell-cell contact here, again here, again here. So these cells are lacking desmosomes, are lacking the cell-cell contact to the epithelial cells, which are suggesting that these pale cells may be mobile cells. They can move on their own. They can be mobile cells, as we are going to see later. So the second step, or the second character, we have seen that there is here interdigitations. Yeah, some things like let's to say this is something like a foot from the right and from the left epithelial cells trying to surround the pale cells. And at many sides, at many slices, you can see that these interdigitations, that these feet are thinning out. So they become very thin, very thin, very thin, until the pale cells have a direct contact, get to be directly contact with the basal membrane of the endometrial glands. So the endometrial glands, so the pale cells try to form a pseudopod. So this is a small foot pushing the basal membrane, until perforating the basal membrane and causing micro rupture. We termed it like ultra micro rupture because we have seen it with ultra microscopy. So this is a transmission electron microscopy, and this is how it looks like on real. This is again the nucleus of the pale cell. This is the cytoplasm of the pale cell. This is the cell border of the pale cell. This is the basal membrane, and this is the stroma. This is a nucleus of a stromal cells. So this is inside the gland. This is outside the gland. To lift down, this is outside the gland, and upper and right, this is inside the gland. And if you see, this is the basal membrane, and it's here perforated by a small foot or protrusion or pseudopod projecting from the pale cell inside the stroma to the outside of the endometrial gland. So following up on these pseudopods, sometimes you see the organelles are migrating from the pale cells from above to the pseudopod, to the enlarged pseudopod inside the stroma. So you see the mitochondria is leaving the endometrial compartment or the glandular compartment inside the stromal compartment, and sometimes associated with shrinkage of the cytoplasm of the pale cells with nucleus migration, and then you have the pale cells outside the endometrial gland. And this is what we have seen here. This is again an ultra microscopy. This is a transmission electron microscopy. This is the basal membrane of an endometrial gland, and to the upper part of the photo shows the endometrial gland. This is the nucleus of a pale cell, and this is the perforation site, and this is the beginning of the formation of the pseudopod. So you have much of the cytoplasm of the pale cells already migrated, already left the bale cell from the endometrial gland inside the stroma. And if you look here inside, you are going to see the nucleus. This is a nucleus has migrated from the bale cell inside the endometrial gland to the stroma. If we have zoomed in more, I think this was 20,000 times zoomed in.
Dr. Mohammed,
yeah.
I will ask the colleagues to close the microphone, please. Yes, yeah. Please, you can proceed.
Yeah. Okay. So just to zoom in inside this site of perforation of this micro rupture of the basal membrane of the endometrial gland, you see this is a continuity of the basal of the cell border of the bale cell with the outside part of the pseudopode. This is the cytoplasm, and this is a second mitochondria, preparing itself to cross over the border to get outside the endometrial gland. So in conclusion, we have added to the theory of the pathogenesis of adenomyosis that we have pale cells. We have an evidence of tissue trauma. This is very evident. So we have an evidence of tissue trauma in adenomyosis uteri. But what is the cause of the occurrence of the adenomyosis? Actually, we think that is the pale cells, these pale cells. This is a cross-section in the uterus. This is the endometrium, characterized by the endometrial glands, and this is the endometrial epithelial cells. And the second layer, this is the myometrium, the muscular layer. And these pale cells situated eccentrically on the outside of situated inside the endometrial glands. They are capable of traveling through the basal membrane of the endometrial gland and going or migrating towards the myometrium and initiate the occurrence of or the building of ectopic endometrial glands inside the myometrium. This was our results, or this was our theory, but I have added also here a bidirectional migration of pale cells. That's to say these pale cells can migrate in two directions. The first direction downwards and through the stroma into the myometrium to reach the myometrium and to form the adenomyosis. But they can migrate also towards the lumen of the endometrial gland and reach the inner layer, the cavity of the uterine cavity, the cavity of the uterus, and migrating through the fallopian tube and settling inside the abdominal cavity to form the endometriosis. And this was actually the work of another group, 2009. They have seen these pale cells and described them just shortly for 3-4 sentences in their publication, that they have seen pale cells inside the peritoneal endometriotic lesions. But this was the first time to describe the pale cells inside the adenomyosis patient. So it was the first time to describe the migration of these pale cells inside the myometrium. We published our results in the fertility sterility, ultramicrotrauma in the endometrium-myometrial junctional zone, and pale cell migration in adenomyosis. In the results or the whole experiment I have done in the endometriosis center at the Charité University of Medicine. This was 2015, 2017. We came out also again with another publication of reproductive science with the Myofibroblast and as an evidence of chronic tissue trauma at the endometrium-myometrial junctional zone in patients with adenomyosis, again coming also from the endometriosis center at the Charité University of Medicine. For this presentation, for these results, I got also the best prize for the basic research on the World Endometriosis Congress 2014 held in Sao Paulo. So the results were very highly accepted and encouraged. Even in the publication or in the volume where our publication was published, there was a commentary from the editors of the journals and saying, is the pale cells the way to go to understand adenomyosis? And the work from the Berlin is a step in the right direction, and we shall be interested in further characterization of these cells. So they encourage so the further research in this way, because this was very promising. At this point, I have finished actually my talk. If you are still interested, if you still have energy, I can proceed with the story continues. If not, it's no problem. I think it's a lot of information inside this talk, and ready to have your questions as well.
Thank you so much, Dr. Mohamed. It was really interesting to hear more about endometriosis from the medical point of view. So I will just ask if anyone has some questions.
From my point of view, I want to know if there are any serological marker for endometriosis. Can we test this disease in patients' blood or serum?
Yeah, this is a good question, but unfortunately, we don't have a special biomarker for endometriosis till now. We have a lot of works. I think Dr. Norhan can tell us about it, from Martin Gutte, University of Münster, with whom I worked also a lot, and do the second part of research about the pale cells also in the University of Münster. We have a lot of microRNAs to try to, I think, not to diagnose endometriosis, but I think we are on the way. But I think it's going to take some time until we can reach a combination of estimation, a combination of microRNAs which can be measured in the blood to save the patient from the operative diagnostic.
Okay, thank you.
Dr. Mohamed, I have a small question about the pale cells, because it's quite interesting to know more about the molecular characteristics of these cells. So, do you know, is there any published data about the gene expression, especially in these cells? Is there any interesting thing to know about them or not?
Yeah, very nice question. Actually, when I arrived in Germany in the Endometriosis Center at a charity, I got from Professor Mixner. So a research proposal, it was something like 15 pages. This was supposed to for a PhD student, not for an MD student, not for a clinician. But I was very, very interested to do it. And the ultramicroscopy and the transmission electron microscopy was only one step inside this research, beside immunohistochemistry in vitro culture. And it was what's called laser isolation of the cells and expression and the gene expression of single cells, and also tissue clamping or cell clamping. This was another part of the research. A very hard work. If you read the basis, it's very, very difficult. But so the story continues. As you wrote this one, I moved to the University Hospital of Münster, and we tried to characterize these cells because they are single cells. So first with the ecadherine, for example, they were ecadherine negative. As you see here, some cells very pale, again chromophobic, are not stained with the ecadherine, so lacking cell-cell contact. They are Musashi-1 positive, and this is some sort of stem cell marker. And we have co-localized ecadherine negative with Musashi-1 positive cells, so we could say, okay, the pale cells are ecadherine negative, but again, they are Musashi-1 positive. CD15 immunoglobulin, it was not so highly specific, the staining, but somehow the pale cells expressed also CD15. This is another marker of stem cell marker, and using the immune fluorescence. So it's a long way to characterize these pale cells. And furthermore, I told Martin Gutte at this time, we have seen the pale cells with electron microscopy. So we have to characterize them using the immune gold labeling of pill cells. And this is a very difficult task. First, to find the pill cells in the new samples. The second one, to establish a protocol for immune gold labeling. But actually, we have reached some results with good results, and this was the immune gold labeling of the pale cells using the Musashi one. So this is the first time to characterize the pale cells with Musashi one antibodies, but using the transmission electron microscopy. For clinicians who are hearing us now, there's a lot of work. This is a very hard work to get these results. What I have explained to you in 30 or 40 minutes, I have spent almost 4 years working on this project to get these results, and this is the whole team behind this. I was the main investigator, but alone I couldn't reach all these results. So this is the whole team working behind this research. Yeah.
Okay. Thank you so much, Dr. Mohamed. I will just ask once more if there is there any more questions? Okay. Yeah. Thank you so much. Thank you so much for this interesting talk, and thank you for the launching of our journal club, GESR Journal Club, and we will just meet next month, the first Sunday in each month, and I hope you can just join us next time. Thank you so much, Dr. Mohamed.
I am going to join you for sure for the next journal club, for the next coming journal clubs. And thanks again for the organization. Thanks again for the invitation. I feel honored to be the first speaker on this or the launching of the scientific journal clubs. Thanks, Dr. Khaled. Thanks, Dr. Ahmed. And I hope I was a good talker for you, or you could understand some of these points, because they are very, very complex, actually. But many, many thanks for your invitation. Yeah?
Yeah, thank you so much. Thank you.
Yeah, okay.
Thank you, everyone. Yeah.
Thank you.